Let's move on to part 3, resting membrane potential. And this is where we go back a little bit to chapter 5. So you should look in your book under chapter 5. So the first thing we want to think about is just about electricity, about charge, about positive ions and negative ions that are going to attract each other or repel. So in this diagram here, we have a positive and a positive charge that's repelling, a negative and a negative charge that's repelling, and a positive and a negative charge that are attracted to each other. So what that does is, if we had an ion in the body that was of one charge and another, they're going to attract each other if they're of opposite charges. So big picture, I'm reading now, electrostatic force, like charges repel, opposite charges attract. Now, it turns out that work can be done by moving charge. So moving charge is considered an electrical current. That's what you think of as current. In your house, you have current in order to get power. So it's like moving electrons in a wire. And that's what we're going to be doing in the body. You're going to be moving electrons. And if you move electrons, you're going to get charge. And that's how we send signals in the nervous system. This diagram below is electrons moving down something. In this case, we're going to say it's a cell, it's an axon, and that's what's going to determine current. And that current is measured in amperes. When we're talking about organisms like ours that have very tiny amounts of charge, it's going to be milliamperes. So M-amp is milliamperes. So if you separate charges and you were to put lots of these positive and some negative charges on one side of a membrane, and then a bunch of different ions with different charges on the other side of the membrane, they're separated, and you're going to keep them that way even though they would like to diffuse. You have many potassiums here that'd like to diffuse out. You have many sodiums out, and they'd like to diffuse in. But if we keep it that way for a little bit using ATP, you're going to create what we call a potential difference. You have a certain amount of charge on one side, a certain amount of charge on the other, and you're keeping it that way on purpose. That is measured in volts, the difference between area A, let's say, and area B. And you're creating that gradient of these different ions. So in this little picture down here, notice that you have a concentration gradient for sodium, meaning, look at the drawing, it's really nice as a triangle, meaning lots of these sodiums outside and so very few, here we have one, inside. So there's a gradient going from many to few. Chlorine is also that way. We have many here going from many to few. And potassium's the opposite. We have many of these potassiums going to very few here. And this takes energy. You're going to keep it that way within the cell to create what we call a resting membrane potential. So this is a crazy picture of static electricity. What this child has done is rubbed her head against the tent and created this amazing static electricity. And the hair of this little kid is all on end. So what actually happened? What is it when you do that? You can do it with a balloon at a birthday party, right? At a kid's birthday party is classic. So what happened here is that the electrons moved away. They moved from the hair to the tent. When you rub on it like that, those electrons are going to move off and jump onto the tent. Now what you have left behind are lots of positive ions in that hair, and they're going to repel. And if they're going to repel, they're going to pull that hair in opposite directions as they try and get away from one another. How far apart can they go? Well, they can go pretty far because this hair is really nicely on end. So those are electrical signals, and there's electrical signals in our bodies and even in plants. So animal cells use electrical signals. Our heart is a really nice example of electrical signals sent in order for it to contract. Plants use these electrical signals, and this is a diagram, a photograph of aloe vera. And if you actually stress it in the heat, it produces a certain amount of charge. You can actually measure that charge if you have a gauge that's small enough to be able to measure in millivolts. So we can measure that. We can measure when there's charge, when there's electricity in something. And so what we're measuring then is a difference between location A and location B. And when there's a difference in charge between those two locations, we call it a membrane potential. So in speaking of cells, the charge between the inside of the cell and the outside of the cell has a potential because the number of positive ions inside and out is not equal. The way we measure this is on the inside of the cell. We get a probe just like you would measure to see if there's any charge coming out of your outlet in your house, and you could get a voltmeter there. You could do that with a cell. You could put a probe inside the cell, and then the second probe from your apparatus is going to be on the outside of the cell, and you're going to see if there's a difference in the number of ions in and out. And we measure that again in millivolts. If we had electrical equilibrium, and this is potentially a cell, and we had equal numbers of positive and negative ions in and equal numbers of positive and negative ions out, then there would be no gradient. If, however, we would move some of those ions out with ATP, let's say, we take it out of equilibrium and we push them out. We talked about this before, the disequilibrium, right? So then we form a gradient. And if we remove enough of those positive ions, we will end up, as we measure the cell, we're going to measure it as being more negative as compared to the outside. So that is the potential difference. So if we could take ions and pump them out of a cell and remove some of those, we could create that resting membrane potential. So human neurons, we know, have high intracellular concentration of potassium, like I showed you when I showed you the picture with the triangles. Intracellular concentration of potassium, high, lots of it, we keep it inside. Low intracellular concentration of sodium, we pump the sodium out. So we keep the sodium on the outside, we keep the potassium on the inside. There's something else that influences that charge, and it's a huge influence, and that is large proteins that are within the cell. Large proteins that have to do with all sorts of metabolic actions that happen within a cell, and those are negative, and they are big. They do not cross the membrane. They will stay inside. So those are going to definitely help in that negative charge, right? Because when we talk about potassium being positive and sodium being positive, it doesn't make any sense that you're making any difference by moving these ions around. But inside the cell, there are these very negative proteins, so they will be negative. Now, if you remove more positive ions from the inside, that cell's going to become even more negative. How do we study this? So this is a giant squid. It's a crazy calamari. So Humboldt squid axons are really big. They're like the size of a little spaghetti, and it makes it easier for us to be able to put a very fine needle-like probe inside and then compare it to the outside and see what's going on. So they have big axons in relationship to their body size, because we're big too, yet we don't have axons that are so big. How would you count these? How do you count the electrons? How do you count the charge against the membrane? Because really it's not an absolute charge. You're not counting total numbers of negative in, total number of negatives out, and then making a difference. You're going to set one of those as 0. So look at the diagram. If you set that extracellular fluid here as 0, so we're gonna give it isn't 0 because it has some negatives and positives, but we're gonna say, okay, this is our standard 0, and then the inside is in comparison to that 0. How different is it from that outside that we're calling neutral? Okay, so it's relative. I'm telling you, the charge across that membrane is relative. This is how you would do it in a saline bath. You would have a cell, and this is a voltmeter, and you have one electrode that's inside the cell here, and one that's on the outside in saline in a water solution, so that you're making a comparison between in and out here. And this ground or reference electrode is placed in the bath and given a value of 0 millivolts. If I give this a value of 0, this is going to be in relationship to this 0 value. Hard to write with this. So what is the resting membrane potential of a real cell? Well, we'd have to measure that. We know that the resting membranes are more permeable to potassium than to sodium, so potassium will leak out more easily than sodium will come in. So potassium is the one with a greater influence. The resting membrane potential is more negative inside the cell, I told you, because of those proteins that are inside the cell. So whatever number we give to the cell is going to be negative. It's going to be minus something. Well, when you measure that difference, it's about 70 millivolts of difference between the inside and the out. And given that we're measuring the negative part being inside, we're going to call it negative 70 millivolts. That's what resting membrane potential of a cell is. It's not sending a message. It's not sending current. It's not connecting to another cell electrically. It's just sitting there. Well, how do you keep it that way? You don't want it to equilibrate, correct? Well, this is maintained by the sodium-potassium pump. So in order to keep it that way, you're going to be sure that if sodium were to come in, we need to pump it back out again. And if potassium were to go out, we're going to pump it back in. And this takes ATP. This is the classic sodium-potassium pump. And the interesting thing is that that carrier there transports 3 sodium out, 3 atoms of sodium out for every 2 potassium that it brings in. It's just unequal in number, but that's the way it works. And I do have videos for you to watch in the other section of my Canvas. So ordinarily, you keep transporting those sodium ions back out and the potassium back in. It's sort of a non-stop event that goes on for it to continually keep that resting membrane potential at negative 70 millivolts when at rest. That's the end of this section. We'll continue on. Thanks for listening.